Hello and welcome back, team. This is Dr. Chriselle Nagallo, adjunct faculty with the College of Science, Engineering, and Technology with Grand Canyon University. In this video, we will discuss aspects of specific immunity. We will cover the significance of the major histocompatibility complex, as well as the differences between passive and active immunity. We will also cover humoral immunity as well as cellular immunity, all of which are part of the third line of defense, which falls into the category of the specific immune system. It is worth summarizing the main players of the immune system, specifically specific immunity. They are your T lymphocytes, B lymphocytes, and your three main categories of antigen presenting cells, which will work to activate your helper T cells. T cells are the same thing as T lymphocytes. There are two main types we'll talk about, CD8 and CD4, which also have specific names we'll cover in a later slide. They're called T lymphocytes because they mature in the thymus, which, if you recall, is the endocrine organ that's present in our pediatric patients, which then atrophy as they start the process of puberty. There are also B lymphocytes. They mature and are significantly produced in the bone marrow. Hence, they are called B lymphocytes. They're also referred to as your B cells. And the main end function of B lymphocytes is to produce antibodies, which we will discuss in more detail as part of humoral immunity. Our three main types of antigen presenting cells are your macrophages. They are monocytes when they are traveling in the bloodstream. When they leave the bloodstream to enter surrounding tissue, they are the same structure, they just take on a different name. And in that scenario, they're called macrophages. Your second main type of antigen presenting cells are your dendritic cells responsible for pinocytosis. So as opposed to phagocytosis, they will drink a little bit of that extracellular fluid. Then there are your B cells. So certain B lymphocytes, certain B cells function by receptor-mediated endocytosis. Here we need the presence of a receptor to drive that intake of extracellular fluid and its components into the cell. That's the main difference between B cells and macrophages, which primarily function by phagocytosis. So there are two main types of specific immunity. One is cellular immunity mediated by T cells. Here, they directly attack and destroy the contents of what's inside an infected cell. And then there is humoral immunity. Here, we produce antibodies courtesy of B cells, and they tag infected cells for destruction. And this is the whole concept behind the vaccination movement we will discuss by the time we are at the end of this slide set. So antigen presenting cells will literally present an antigen, which we've defined before, but we'll define again because it is an important part of this discussion. Antigens are a certain part, a specific protein or a segment of a protein of an infectious agent. And when they come from an infectious agent, aka a pathogen, they become known as an epitope. So there's a whole process outlined for you here in the schematic diagram. It begins with phagocytosis. And then the pathogen passes through the lysosome. The lysosome breaks down the pathogen into its protein components, among other things, and then it presents that protein or part of that protein as an antigen on the surface of the cell as part of the major histocompatibility complex, often abbreviated MHC. And at that point, it's become known as an epitope. And this becomes really important because this is the mechanism by which the outside of the cell, so basically other players of the immune system, this is how they'll know whether a cell of the body is infected or not. We cannot, without breaking the cell membrane, because when that happens, that human cell will also be damaged, and that presents significant ramifications. So there are two types of MHC complexes. One is present when a human cell is infected. We just discussed that. That is all part of the MHC1 complex. And then the second presents only if you are an antigen presenting cell. So who are those again? These are your macrophages, your dendritic cells, and then your B cells. So it's worth knowing, and we'd like to ask about exceptions, that all human cells in the body will present an MHC1 complex, except for red blood cells. And this explains why infections of the blood present differently than other infected cells in the body when it comes to clinical presentation. What we'll now do is match the MHC1 complex and MHC2 complex to the player in the T lymphocyte arena that will become activated as a result of MHC1 and MHC2 activation. So let's define the T lymphocyte players. You've got two main types worth knowing really well: cytotoxic T cells, often abbreviated TC, and then your helper T cells, often abbreviated TH. Now, when we first identified these T lymphocytes, we noticed that they presented with different proteins on their cell membrane surface. CD8 is what we found on cytotoxic T cells, and so they're often synonymous with one another. And then CD4 is what we found on the surface of helper T cells. And so CD4 is synonymous with TH, is synonymous with helper T cells. One of the most challenging parts about immunology is recognizing that there are many ways to reference the same cell. And so it is worth knowing and being very familiar with the different ways in which one specific cell can be referred to. And then this next slide is a summary, really helpful overview of how the MHC complexes interact with their T lymphocyte counterparts. So when MHC1 is activated, it will recruit CD8, also known as T cells, to confer a specific action. And then when MHC2 complex is activated, then CD4, also known as your helper T cells, will then be activated. In the next series of slides, we will discuss the specific mechanism and physiology by which this occurs, starting with MHC1. So, as you recall, MHC1 will present an epitope on a human cell when it is infected by a pathogen. What then happens is this epitope will draw in or attract a cytotoxic T cell, also known as a CD8 cell. Through almost a lock and key model, the CD8 protein is found near this complex that's formed between the MHC1 complex on the infected human cell and the T cell receptor on the CD8 cytotoxic T cell through a process called co-stimulation. When this occurs, when this entire complex forms, two main events occur. One is the release of perforins, which we saw before in the context of natural killer cells. They will literally perforate the plasma membrane or the cell membrane of the infected cell. What will also occur is the release of lymphotoxins. So chemical agents that serve as toxins that come from the T lymphocyte. And perforin release combined with lymphotoxin release have the end goal of destroying the infected human cell, because if they are infected, then the pathogen has the capacity to hijack cellular metabolism of the infected T cell and increase exponentially, which can have significant negative ramifications on the human body. So by destroying this infected T cell, let me backtrack, this infected human cell, then we're nipping the problem earlier on. Yes, we sacrifice a human infected cell, but it is a small price to pay in order to protect other human cells from potentially becoming infected by the same pathogen. So the bottom line here is that MHC1 meets the CD8 cytotoxic T cell. The antigen presenting cells, which again are the macrophages, dendritic cells, and B lymphocytes, when they engulf a pathogen, they present an epitope on the plasma membrane as well, but for a slightly different purpose. In this case, the MHC2 complex presents an epitope in order to recruit helper T cells, also known as CD4 cells. They all mean the same thing. There are in the body inactive T cells and active T cells. If there is an active infection, and we'll know because MHC complexes will be activated, then the goal of the active T cells is to become active to help out in this war against the pathogen. So we're going to take the macrophage as our main example here. The macrophage will engulf the pathogen through a process known as phagocytosis. It will present parts of the engulfed pathogen in an epitope as part of the MHC2 complex. The presence of the MHC2 complex on the infected antigen presenting cell will attract T cells, which have a receptor, as well as the CD4 protein on the cell membrane. These are the helper T cells. What the helper T cells will then do is activate other T cells. So there is they basically provide a call to recruit even more cytotoxic T cells. These are your CD8 cells. And then they also activate B cells, which will then mature into plasma cells, which will then activate the production of antibodies, which leads us to our discussion on humoral immunity. The end goal of humoral immunity is to produce antibodies. Antibodies are proteins, the big globulin that has specific for a specific certain pathogen. Here we overview the entire process involved in order to make antibodies. B cells will differentiate into plasma cells, and plasma cells will produce and secrete antibodies. Here is the classic Y-shaped antibody structure, worth knowing really well. There are 2 heavy chains and 2 light chains structured in a Y letter. At the top of the Y are 2 antigen binding sites. These are specific to a certain part of a pathogen. Could be bacteria, could be a parasite, could be a virus. The heavy chains fall into categories that will dictate the class of antibody. If you recall, we talked about complement binding sites in a previous discussion. They will bind here to communicate with antibodies when it's time for these to be activated again after a secondary exposure to the same pathogen. Antibodies will go through a process of opsonization, which correlates back to our discussion on the complement system involving C3A and C4A, which will then bind to the complement binding site on the heavy chain. There are 5 main classes of antibodies dictated by the structure that's on the heavy chains here, here, and here. IgG, IgE, IgD, IgM, and IgA. IgG is the largest and most diverse class of antibodies. This is the main player in a secondary exposure to the same pathogen. IgE is typically what is produced and secreted in an allergic response. This is the issue in an anaphylactic reaction. IgD is what's produced on the surface of B cells and is involved in the sensitization of B lymphocytes. IgM is the class that is secreted first, followed by IgG. IgM is the antibody that we will detect in a more recent exposure to a pathogen. IgA is the antibody class that's found in glandular secretions such as mucus, tears, saliva, and semen. IgA antibodies are the class that can be transferred from one individual to another because glandular secretions can easily do that. The whole point of antibodies is for the process of agglutination. We've discussed this before in a different conversation. In that context, it was in the context of the ABO blood type system. Here, same concept, slightly different context. When the antibody meets a specific antigen that's on a pathogen, it will result in a process called agglutination, which will ideally result in the destruction of that pathogen. The immune system is an intricate play of different players that come together to eventually ensure that the body has the protection it needs to withstand a potential infection. This is an overview diagram of how the different players come together, which I will leave for you to review. In this next segment, we will discuss the difference between a primary and secondary immune response. Primary immune response involves the sequence of events that ensues after the initial exposure to a pathogen. A secondary exposure occurs later on, in which the body will activate memory cells that formed towards the end of the initial exposure, so that if we saw the same pathogen again, we have better defenses, more efficient tools to deal with the infection the second time around when compared to the first time around. This is the whole concept behind the vaccination movement. I want to highlight these sets of diagrams, these graphs here, because they say a lot. In the primary response, IgM class of antibodies is what will be seen first, followed by shortly the IgG antibody class. If after, let's say, 2 years, the same pathogen was seen or detected by the human body, then the secondary response will initiate IgG antibody production first, because we've had memory cells that were produced after the primary response that were ready to make these antibodies specific to the pathogen so that we can take care of the infection faster and more efficiently. In the classical definition of a vaccination, an antigen, deactivated virus, or a weakened form of the pathogen was introduced by way of a syringe and needle that was designed to activate long-term immunity. So, MHC complexes, I apologize, not MHC complexes. The end goal of vaccinations is to produce antibodies from B lymphocytes, which differentiate into plasma cells, but in such a way that the pathogen load does not overwhelm the immune system. So just enough to get the antibody production, but not enough to cause a debilitating illness. So later on, if the patient saw or was truly exposed to that same pathogen, then memory cells will relatively quickly produce and secrete IgG antibodies to that pathogen to avoid, to fight that pathogen and avoid long-term illness. And that brings us to our last discussion, last topic of this video, and that is the difference between active and passive immunity. Immunity can occur naturally or artificially. One can argue that active immunity is the best, most efficient type of immunity. Here, the patient is exposed to the pathogen in some way, shape, or form. It stimulated the patient's immune system. And the patient was also able to recover from the infection. Vaccinations would fall into this category. In passive immunity, there wasn't a true infection. Rather, the patient received antibodies so that if the patient was exposed to a certain pathogen, then given that it is not too much time has passed, then the antibodies could confer their action and provide some sort of immunity short term, but effective if exposed within that expected window. Maternal immunity falls into this category, and this explains why the CDC and WHO does recommend breastfeeding only for at least the first 6 months of an infant's life, because not only does breast milk have a specific nutritional blend for infant, but it also offers antibodies for infant that's passed through breast milk, and it also passes through the placenta. There are other forms of antibodies given in an injection format, different from a vaccination, because of the component that's actually being injected. These medications require the injection of antibodies to produce short-term immunity. Specific examples are beyond the scope of this class. A quick note about allergies, because it's common and we're entering into allergy season. Allergies is essentially the immune system going haywire unnecessarily. So the immune system thinks that pollen or certain aspects of the environment are potentially detrimental to the body. And so IgE receptors on mast cells and plasma cells will activate a response, which typically involves histamine release. And if you think about what histamine does, it explains a lot of the symptoms that are present in an allergy response, especially an allergy response to seasonal allergies. This is why an antihistamine, like Claritin, will provide symptom relief for patients who suffer from moderate to severe allergies during allergy season. There are several slides that give you more background to the major histocompatibility complex, which I am more than happy to answer any questions about at a different time. Thank you for tuning into this video. I hope you find it very helpful.